Why does it matter to grasp the concept of polymerase chain reaction, or PCR as it's commonly called? Well, it's not just a staple in the world of molecular biology, but also a vital tool in medical and biological research. It's like the bread and butter of DNA manipulation, so buckle up for an enlightening journey into the world of PCR. Picture a scene from a crime thriller where they find a single strand of hair at the crime scene. How do they get enough DNA from that strand of hair to identify the criminal? Enter PCR, the ingenious process that allows scientists to create millions of copies of a specific DNA sequence from a tiny sample. Now imagine a zipper. That's your DNA strand. The PCR process starts with denaturation, where the DNA strand is heated to about 95 degrees Celsius. This heat unzips the DNA strand, separating it into two single strands. Then comes the second step, annealing. Picture a winter's day, it gets colder, right? That's precisely what happens in this step. The temperature drops to 50 to 65 degrees Celsius. This allows the primers, which are short strands of DNA, to bind to the DNA strand. Next, we have the extension phase. This is where the magic happens, where the DNA gets duplicated. The temperature increases again to around 72 degrees Celsius. A special enzyme called Taq polymerase starts adding nucleotides to the DNA strand, creating a new strand of DNA. And voila, you have a copy of your DNA. But we're not stopping at just one. This process repeats around 30 to 40 times each cycle, doubling the amount of DNA. So by the end, you have over 1 billion copies of the DNA sequence. It's like having a Xerox machine for DNA. So why is PCR so important? It's simple. PCR is the backbone of many scientific breakthroughs. It's used in genetic testing, diagnosing diseases, forensic science, and even in researching ancient DNA. Without PCR, we might still be in the dark in many areas of biology and medicine. In essence, PCR is a powerful tool that allows us to amplify a specific DNA sequence from a tiny sample. It's a process that involves heating and cooling the DNA to allow it to separate, bind with primers, and then extend with the help of an enzyme to create new strands of DNA. This cycle repeats numerous times, leading to billions of copies of the DNA sequence. So the next time you hear about a breakthrough in genetic research or a criminal caught through DNA evidence, remember the unsung hero, the polymerase chain reaction. It's not just a process, it's a revolution in the world of science.